안녕하십니까. 지금부터
Good afternoon.
BIOPLUS-INTERPHEX
Now we will begin the BioPLUS-INTERPHEX 2022 Biomimic Technology Beyond the Borders of Animal Study and Clinical Trial session. My name is Ko Sung-ho. I will serve as a chair and speaker, and thank you very much for your time. Then we would like to begin the session now.
세션의 이제 개요를 간단하게
I would
말씀드리면
like to give you the overview of the session.
신약을 개발하는데 약
Now,
10년
when we
정도의
develop
엄청난
new
시간입니다.
drugs, it takes about 10
국적으로
years
2조
and more than 1 trillion to 2 trillion of cost.
지금 그럼에도 불구하고 우리가
Despite these
많은
time and
지금까지
efforts,
신약 개발을 위해서 많은 동물
still,
실험을 하게 되는데요.
we need to go through the animal study, and it was inevitable that we need to have the sacrifice of the animal. To overcome this problem, new technologies are coming up. And one of the technologies we pay attention to is biomimetic technology. And I believe we can see the present and the future of this technology. So I hope you can be inspired by this session.
그러면 지금부터
Then
각각 오늘의 네 분의 연사분이 발표할
we will have 4
첫 번째로
presentations,
제가
and I will serve as the first presenter.
저는 휴먼 에이스 대표
My name is Ko Sung-ho. I'm the CEO of HumanAce, and I'm a bioengineering department of Chai University
창업을 해서
professor, and I also established my own company.
회사 발전을 위해서
And for the development of the company, I'm making lots of effort. The title of my presentation is Organs on Chips for New Drug Development. I will serve as a presenter and speaker
간략하게 제 소개를
and chair. So I would like to introduce myself briefly. And
엔지니어링과에서
in Missouri University,
기술로 박사학위를 받았습니다.
I received the PhD, and it's been about 11 years since I joined the Chai University. Then I would like to begin my presentation. My company is HumanAce. My company was established 8 months ago in 2021 October, and we have 8 members, and most of them are researchers. And we also have good financing experts, so we are preparing for IPO. And for new drug development, our goal is creating the world without animal test.
그래서 결론적으로 우리 회사는 이제 장기를
Our company is trying to use the organ on a chip. Commercialization is the ultimate goal.
그래서 지금 우리가 신약 개발하는데 대부분의
Now, when we develop new
실패를
drugs, 90% are ended up with failure. So when we try about 10,000 candidates, only one is successful. So there are 2 reasons. The first one is a preclinical study. We went through the 2D cell culture process, and our have ECM, including extracellular materials, and they have fresh oxygen and vascular cells, and the waste will be also excreted through the vessels. But 2D system will not reflect this real situation. So in a clinical study, the outcome is totally different. Another one is the animal test. Animals have different DNA and genes from human being. So according to the FDA data, the mismatch of the animal test and clinical study is almost 92%. Only 8% is matching between the animal test and human test. So we need to move away from the animal test. That's why we are studying about organ on a chip. And the focus product we have is heart on a chip. It is mimicking the heart of human being, and we are developing the organ on a chip. And we also develop brain on a chip, and we completed the development. And another product we are focusing is liver on a chip, and we almost finished the development. I have very short time of 15 minutes, so I cannot present everything. So I will just focus on the heart on a chip. With this heart on a chip, we can see the efficacy and toxicity of drugs. When we develop new drugs, efficacy test is important, but toxicity test is as important as the efficacy test. So there are many candidates, but they have been withdrawn because of the cardiotoxicity. It takes up about 45%, and it is followed by liver toxicity. Almost 80% of the candidates have been withdrawn because of this toxicity. That's why I'm focusing on the heart toxicity to conduct my study. And our goal is not using the animal cell, so we need to use the human cell. However, there is no way we can secure human heart cell. So if we use the cadaver, then they are not beating. So we need to harvest the stem cell, and it can be differentiated into cardiomyocyte. There are various shapes, and when we conduct the experiment, we identify the heart-shaped cells. So once it is differentiated, then they will start beating. With these cells, we use the 12-well plate. In a 2D situation, we measure the beating per minute. Please click the slide. Now, on each well, we have a different BPM. Sometimes we can observe 140 BPM, sometimes it is 70 BPM. So it is irregular, it is arrhythmia. And even though they have been differentiated from the same stem cell and they are on the same well, but beating patterns can be different. So when we test the toxicity on these cells, it is not correlated because they are not mimicking the important cells in our human body. So we need to have a platform that can overcome this problem. That's why I carried out the activities. In the heart structure, I paid attention to this structure. This is a pyramidal structure from the cardiomyocyte. There is a collagen, and blood vessels provide fresh nutrients and oxygen. So this pyramidal structure is very important. This is the actual photo of the microscopic picture, and we first lay down the PR plate, and we also used the film mask and exposed it to the UV, and multiple polymers will be added to it, and after drying we separate them. Then it is turned into heart on a chip, and cardiomyocyte can be injected into this. Please click the picture. Then cardiomyocyte, differentiated from the stem cell, will be injected. And we can confirm that they grow very well. And in a stable manner, Please click the slide. We can observe the beating at 70 BPM. What is important here is there might be plus minus 5 BPM. They have consistency and they have regular beating. So based on this, we can say that we can test multiple drugs and we can enhance the reliability of the test data. We are not trying to write the paper here. Rather, we would like to commercialize the drug. And the consistency is very important. Otherwise, we cannot successfully commercialize them. So consistency of the data is very important. And now we can have reliable data. So in order to confirm the quality of the organ on a chip, we use the positive control drug. And one of them is isoproterenol. And in a clinical situation, it enhances the beating of the heart. So baseline is 75 BPM, and once the drug is injected, then the BPM will increase to 105 BPM. And when we see the motion tracking, in the baseline, the BPM was 75, but once this drug is injected, the beating is even stronger and it is faster. We can see the outcome in the motion tracking as well. And there are also drugs that can slow down the beating. One of the examples is nifedipine. And we also confirm whether the heart on a chip works in the same way. So the baseline is 65 BPM, but once the drug is injected, then the drug will work so that the BPM will slow down to 43. And once we inject more drugs, then the beating will even stop. So we don't have to use the animal test or in vitro 2D test. With this platform we developed, we can enhance the chance of success. And we also monitor the motion tracking, and we can compare the control group and after the injection. After the injection of the drug, the heart beating will be slowing down and it is weaker. And brain and heart is very important to measure the electrophysiology. So not just about the beating, cell viability, and once a drug is injected, we need to monitor how electrophysiology is changing. So on one chip, we can measure all things. In order to create this kind of organ on a chip, we also manufacture the microelectrode array, and we wanted to have the perfect alignment of the channels on the chip. So depending on the injection of the drug, we measure the field potential to measure accurate efficacy and potency of the drug, and we also can measure a heartbeat rate, contractibility, and viability assay. So the black area is the electrode that can measure the electric signal, and we also injected the materials differentiated from the stem cell, and after incubation, they will start beating, and isoproterenol or other drugs will be treated. Then, as just like the ECG, we can measure the efficacy and toxicity. The blue line is control group, and once isoproterenol is injected, then we can see the green line. And if we inject the nifedipine, then it will show red line.
임상 데이터와 거의 똑같이
and it is almost similar to the previous clinical data.
심장의 효능과 독성 테스트에 있어
So in the
피지올로지를 측정하는 게 굉장히 중요한데
efficacy test and toxicity test, electrophysiology is very important. So we already completed the development of this chip.
그리고 우리 지금 회사는 좀 처음에 말씀하셨다시피
As I mentioned before, not just a heart on a chip, we are developing brain on a chip, and we can conduct toxicity test, and we can also study Alzheimer's disease or Parkinson's disease that is related to the degeneration of the brain.
그다음에 이제 NA 그 FLD와 같은
And
비알코올성 지방산 지방 모델
non-alcoholic
연구가 끝나고
a disease such as NAFLD can be studied on a chip.
지방간을 치료할 수 있는 혹은
So fatty liver can be studied, and we can identify the candidates for fatty liver drugs.
그다음에 우리가 장기들은 간을 통과해야 되기
And
때문에
all the drugs need to go through liver and heart.
장기들이 연결되어 있는
So human body have multiple organs. That's why we are trying to develop
여러 가지
multi-organ on a chip. And
그 TME, tumor microenvironment를
we would like to mimic the microenvironment of the cancer by creating the cancers on our chip. And cancers are related to immune system. That's why we would like to develop immune cancer on a chip and microbiome on a chip, because it can affect to multiple immune system, including brain. So we are developing multiple organs on a chip. In the future, all the organs can be connected on a chip. It can be called as human on a chip. It can serve as an avatar. And in the second half of this year, Avatar 2 movie will be released. Just like that, we would like to start the commercialization of human on a chip on the second half of this year, but it will not be possible. So our goal is releasing this product when the movie version 3 is released. And there will be 3 more presentations from organoid side, MapsGen, and TNR BioFab. They're amazing companies. Compared to them, our company is a new company. And compared to these companies, our value is still undervalued. So if there is any investor who are interested, then we are waiting for them. With this, I would like to conclude the presentation, and we have limited time. So we don't have time to accommodate the question now. We will cover them in the panel discussion. And after that, if you want, we can have a one-on-one session. Then we'll move on to the next presenter.
So next speaker will be Yu Jong-man, CEO of Organoid Science, and Professor Yu serves as a professor at the Graduate School of Medicine at the Cha University, and also he is a director of the Cha Organoid Center, and he has accumulated a lot of expertise in organoid sector. And presentation title, actually we did not see the title, so could you please say your title? Thank you. And could you please start your presentation? Can I get a remote controller? Good evening, afternoon. I am Yu Jong-man from Organoid Science, and we have such a huge turnout. So for the interest of time, I will brief you on our company overview and technology. So first, we have 2 business. One is regenerative medicine, and also mainly I'm talking about the platform for evaluation of new drug. So we were established in 2018 in October, and we have employed more than 80 workers, and we have centered Pangyo R&D Center, and also we have a Gwangmyeong GMP Center, and also we have a GMP Center for Seoul Asan University Hospital. And this is a brief introduction of me, and this is the leadership. And actually, we are trying to utilize new science to provide a better experiences based on organoid technology. And actually, we were established in 2018. And actually, we have attracted 8 billion won of Series A funding. And we also successfully attracted Series B, which allowed us to carry out research and on commercialization. And we have witnessed revenues, and our revenues are on a constant increase, and regenerative medicine will be administered in the clinical trials, and this is the last phase before we get into the clinical trial. And organoid, well, our brand is specialized in organoid brand, and we have commercialized a lot of products line. So organoid screen is about platform that I would like to address before you. And organoid platform is about organoid regenerative therapy, organoid research is a lineup of reagents required for organoid development. And so let me spend more time on OASIS screening. So the concept of organoid, for the interest of time, CEO Ko mentioned organoid actually chip mimics the human body, but we mimic human body by utilizing stem cell. So we have developed cancer organoid, and we have called this platform Odyssey, and ONC means oncology. So Odyssey is an oncology-related drug screening platform. There are many cancer patients, and they are mainly the donor of the tumor tissue. And we also, with a variety of methods, we validate the characteristics of individual to find disease-related characteristics. So we like to identify the similarity between markers and also the reproducibility of markers. An MSI type will be identified with our platform. And actually, we analyze each genetic information using NGS and MIS analysis, and we find out a similarity. And we have a lot of challenges. So if there is a lot of media, then there will be a lower mutation. For our company, to reduce that, we standardize the protocols and develop few compounds to improve and maintain patent generate information for a longer period. And with organoid developed, we have witnessed the similarity with the patient with an organoid. So we have extruded organoid from pancreatic cancer patients, and we have witnessed the similarity at a level of 80% on our platform. And with this form, there are cancer target drugs can be tested. As you can see, there are many research institutes partnering with us, and we have carried out collaboration to screen the drug candidate for oncology. And actually, the cancer organoid is consisting of cancer cells, so immune cells and tumor microenvironment should be mimicked. So this is not only confined to organoid, but also cancer model has shared these challenges. So in order for us to overcome this, we have utilized cancer cells and immune cells. And T cell target CD8 T cell target drugs and immune checkpoint inhibitors have witnessed a shortage of test platform. So this is why we have developed Odyssey platform. And CD8 T cell and cancer a cell are co-cultured, and so in order for us to mimic this immune system, we have to put much emphasis on the similarity with cancer organoid and patient-derived tissue cell. So we co-culture those two. And as I mentioned before, T cell platform 1.0 is targeting, and there are a variety of modality drugs, macrophage, or tumor-infiltrating lymphocyte, or T cell, regulatory T cell, are engaged. So at an incremental manner, we would like to advance this platform so that this all can be tested via our platform. So we are now in a process of upgrading our platform. As you can see from this platform, this video shows how it works, and the green label is tumor organoids, and the gray small dots are T cells. And after treating with the immuno-oncology drug, tumor organoids die, and the color of the organoids change from yellow to red by adding PI. So we quantify the data on the graph, and this is a simulation of the cancer cell apoptosis. And this is a platform that tests the immunology of a cancer patient. And also combination therapy strategy can be set up by our platform. As I mentioned before, we can add macrophage to test antibody drugs with a complex MOA, and in version 3.0 and in 3.4, regular T cell is co-cultured, so we are able to identify MOA and test the complexity of MOA. And compared to the previous model, we have a lot of broader applications. So we have a lot of collaborating partners. It's early stage. So actually there is no platform for target section in the early stage, but we are identifying how valuable it is to have such a platform in an earlier stage. And also we have focused on identifying a lot of MOAs. And also this is another collaboration case. So we can identify higher efficacy compared to the existing or legacy one. And actually, we have higher efficacy of drug, which was not responsible in the on the other platform. And also we have to gain in vivo results. So we test organoids, but at the same time, at next level, we try to test in vivo via our platform. So we try to humanize immunity organoid mouse model via Odyssey ONC platform. So with this organoid, we can test in vitro and in vivo. We also provide a environment that is very similar to that of human body. So interest time, and I will change my gear to Oasis screening. And going beyond the oncology, let me touch upon several indications. And actually, using our respiratory organoid lineup, such as tonsil, adenoid, and lung organoid, we establish in vivo disease modeling. And also, there might be a next pandemic, and it's very hard for us to model this. So antiviral agent should be developed by testing animals. So in order for us to overcome this barrier, we utilize organoid for infectious disease such as norovirus. So actually, norovirus and COVID-19 and other infectious disease in vitro are very hard to be tested. So we try to provide a variety of organoid infection model. So tonsil or respiratory organoid are created. SARS-CoV-2 coronavirus are injected to this organoid for infection, and also we have do a variety of treatment administration. And with this, a lot of different responses, we identify similar in vivo response. After this modeling, we not only test efficacy but also identify the mechanism of actions of drugs at a higher accuracy. So with utilizing this, not only infectious disease, but also normal intestinal organoid or lactobacillus or other microorganism that is good for our human body can be identified for its efficacy and its impact. So this can be also one of the application of our organoid. And as I mentioned before, our organoid are derived from patient tissue, but iPS and other stem cells are utilized for producing our organoids. Microtechnology is developed for the high speed and large scale production of highly uniform organoids. So this is unmatched in terms of speed compared to the existing legacy organoids. The case in point is that iPS and ES cells are used to produce organoid, and there are complex process here, and after you complete this process, there might be a lack of uniformity, which is very hard for you, which makes difficult for you to use this organoid on the ground. So, but our organoid is varying in size, and microtechnology can be utilized to produce organoid within 30 days, and uniformity is ensured. And also, this platform has a lot of industrial applications, and I will skip through this because of the interest of time. And there are two platforms and developed here. So, brain organoid is produced by microtechnology with a faster time and with a higher uniformity.
Microorganoids are functionally mature. Microorganoids from day 30 of differentiation display dramatically enhanced electrophysiology.
And this is how it is produced, and the produced organoid can be used for modeling. And as you see this is a organoid from autism patients, and this is a modeling by microtechnology. And also the another organoid is a liver organoid. And as you can see, there are varying in size and there is a differences, but liver organoid shows the similarity, a higher similarity. And after you complete this, there is a Kupffer cell. This organoid contains not only hepatic cells and Kupffer cells and other enzymes as well. So previous speaker mentioned non-alcoholic fatty liver disease and other disease can test the drug efficacy based on platform. So our company is utilizing cancer organoid to develop immuno-checkpoint inhibitors and immuno-oncology therapies. So we try to provide a solution and answer the questions with regards to infectious disease, brain diseases, and target anti-cancer therapy. And we are doing a lot of collaboration. So actually, regenerative medicine is the next part. So let me conclude my presentation for the interest of time.
Thank you very much for the good presentation. Then we will have Mr. No Hoon Seok from MepGen. Dr. No Hoon Seok received the degree, the PhD degree with the nanochip related study from Netherlands. And he is developing the organ mimicking technology for the new drug development. Mr. Renault, please go ahead. My name is Ro Hun-seok. I'm the CTO of MapsGen. Thank you very much for the invitation. And I would like to introduce the company and my company's product. It is a good opportunity for me. So thank you very much for this chance. The title of my presentation is Micro-Engineered Physiological Systems for Advanced Pharmaceutical Development. But before my presentation, I would like to talk about my company. Then I will cover a human organ on a chip. We are developing MAPS-X series. This is a tissue barrier chip, and we also have a vascularized organoid chip. And we also would like to share the business model we have in our company. Our company was established in 2019 August, so it's been almost 3 years, and we received the investment from Korea Investment Partners, and in the Series B of last year, Kium Investment Security and Quorum Investment also participated in the funding process. First of all, we had the first pipeline, organ on a chip system platform is we are creating. And we also have MGX series, and this is using biomimicry platform to handle and address the CNS disease. We are developing nanoparticle-related treatment. And in our chip, we are mimicking the BBB, and we are developing nanoparticle therapeutics. And we have the system that can develop these drugs. The CEO is the professor of the U.S. University, and in the postdoctoral process, the professor was also part of my company. So we have 18 researchers, and more than half of them received the PhD. So we have high development capability. I would like to cover the human on a chip. When we talk about human organ on a chip, in development of the new drugs, we always talk about the success rate. When we talk 1%, you cannot see how small it is. Let's say we create 100 smartphones, but 99 of them are discarded without activation. So in a preclinical and clinical study, there will be difficulties. And in order to overcome these difficulties, we are mimicking the human organs. We are mimicking the function of the organs. And what is important is the barrier between the organs and blood vessels. How much drugs can be permeated and how much interaction can be observed. Eventually, when we culture the cells, if we do that in a 2D environment, we need to use the one type of cells. So we need to have the membrane one side and blood vessels the other side. But there is a problem. When we do the culture, there are static and dynamic situations. When we receive the cells and culture them on a membrane, then gene expression will be different because in our human body, there is always the circulation of human blood. So with the circulation of the blood, cells will recognize this and the cells will express the receptors, but they will not be represented in the 2D cell culture. And another problem is throughput. One chip will be a set. For example, we need to operate 96 chips at once. So that was the challenge or limitation. However, in our company, we developed our own technology to overcome this problem. And what I would like to say is we can culture 2D and 3D at the same time. I'll cover this in the later slide. Now we are focusing on BBB, blood-brain barrier. So the blood vessel in brain, how much drugs can be permeated. These are the focus area in our company. And actually, human brain is well developed and it is well protected. So once the drug is injected, it will not be transferred to the brain. In other words, when we inject the drug, it is not permeated. That is the problem for us. So in order to overcome this problem, we created the BBB in the chip. So this is the structure of the chip. The video is not playing, but I will verbally explain this. On top, you can see the blood vessel, and at the bottom, you can see the organ. And in between, there is a membrane, and we included the blood vessel cells and parasite, and other cells will be cultured. In our product, at the bottom, we have gels and cell mixed together for a 3D culture, and we can inject it once again. And we also have the side channel so that we can inject the liquid for culture. Basically, when you look at this, here we can see the parasite, and we can also see the creation of the astrocyte in 3D. And as I mentioned before, we have 3D channel. So other cells, such as neuron cells and other cells, can be injected to create a model. And nowadays, the trigger of the Alzheimer is the accumulation of amyloid beta. That's what I believe. In that case, we can create the model for Alzheimer's disease. We first create the BBB, and we also use the side channel to create the Alzheimer's disease model and inject the drug. And through the blood, we can see how much will be permeated. And after the permeation, how much amyloid beta can be reduced. In Harvard Ingerberg Lab, when it was first announced, we created the BBB as an organ on a chip, and we also created the 3D structure and 2D structure as well. It would be difficult for you to look at the graph, but this shows the in vivo similarity with in vivo when we culture the astrocyte. And when we culture the cells, we are using the nanoparticle drugs, and it is injected into the model. And with a specific receptor, the BBB will be penetrated. We confirmed this already. And 50% were remained in the blood area, and the other 50% have been penetrated and permeated into the brain. And we also inhibited this receptor to see how much is inhibited. And we can see that 80% still remained in the blood area. So we can see the role of the receptor. And we also have developed another product, vascularized organoid chip. The video is now playing, but at the top, we have the blood vessel mimicking structure, and we changed the base structure using flow cussing. We included organoid and we can capture this.
지금 단면을 보시면
When you look at the side way,
이러한 스페로이드나 organoid를 캡처를
spheroid and
하고
organoid can be captured, and with the blood vessel cells,
만드는
new blood vessels can be created,
튜머 타겟의 스페로이드
and this
연결을 해서
can be connected with the tumor target spheroid
할수 있습니다.
to test the target.
이게 튜머 스페로이드가 있고
Now you can see tumor spheroid, and it is vascularized and connected together. This is important because
이제 환자 맞춤형
we
의약품이라고
are
했을 때
trying to develop the
항암제를
patient-customized drugs.
치료를
It
받았을 때
means
저희가
anti-cancer drugs.
그 샘플을 얻게 되면
And we
저희가
receive the
스페로이드를
samples
만들어서
and create the spheroid, and we connect the blood vessels to test the anti-cancer drugs. And there are two types of anti-cancer drugs. The first one is targeting the tumor to treat the cancer. And
그 부분들에 대해서 끊어주는
second
그런 항암제가
drug
있습니다. 그럴
is
경우에
disconnecting the
어떤 약이 얼마큼
vascular
치료될지
connections.
이런 거를
So
저희
depending on
시스템을
the
통해서
anti-cancer
확인을
drugs,
하고
we
항암제
can
칵테일을
confirm
만드는데
the efficacy,
적용을 할 수가
and we
있습니다.
can reflect the outcome into creation of the cocktail.
저희 지금 현재 양산해서 제품으로 출시된
Now
제품은
we've
플라스틱
been
칩으로
commercialized
말씀을
these
드리면
products,
TBC와 VOC 그리고 하이드로프 버전
TBC,
개발
VOC,
중이고요.
and
그래서 그 안에서 이제
HTC
TBC
is
안에서
in
어떠한
the
모델을
development
구성할 수
process,
있는지에
and
대해서 VEB, BBB 그리고
we
뉴런
have
들어간
the
MB,
VEB, BBB,
그리고
NVU,
VOC를 이용해서
and using VOC, we are
ORD
developing ANG,
제품이
TME,
현재
and
판매 중에
ORD. These are already
그래서
available
다시
in the market.
말씀드리면 저희 회사에서는 지금 3D
If I
organ
mention
테크놀로지를
again,
이용해서 플라스틱
by
칩과
using 3D
그리고
organ on a
거기에
chip
상응하는
technology,
모델을 개발 중입니다.
we are developing
거기에
the model
MGX로
and
나노파티클을
nanoparticle represented by MGX. We are developing new drugs. When we combine these two, it can be used as a platform, and this portfolio has high expandability.
지금 서비스는
Now we can provide the services.
올가노나 칩에 사실 올가노나
The
칩을
organ on a
만들었을 때
chip
가장
we
문제점은
created
뭐냐면
have some problems.
셀을 적용을 하나하나를 더 할 때마다
When we add another cell,
속도가 더 높게
it takes more time and it requires more skill, so it is difficult to fabricate.
숙련된
However, as I mentioned, we have skilled workers. They create the chips and receive the drug and apply it to test the drug. We provide the research service, and we are also developing another technology.
BBB를 형성 후에
After creating the BBB, we freeze them to ship them to the end user. Then the end user will thaw the product and inject the drug. We already applied the patent, and now we are in the process of optimization.
다른 회사에서 올가노나 칩을
Other companies are developing organ on a chip as well, and they explain that the chips are ready to use.
소분된 셀이라든지
So chips are frozen,
세포라든지 미들.
and they provide the cells.
여러분이 생각하실 때 이제 밀키트 같은 거죠.
So it's like a meal kit.
요리를 하셔야 됩니다.
Anyways, you need to cook it. However, in our case, you can just thaw it, then you can use it right away.
그래서 현재 저희는 칩 세일즈, 칩 판매를
Now
통해서
we are selling our
그
chips,
리서치 서비스도 같이 하고 있고
and we provide research service.
말씀을 안 드렸지만
And even though I didn't cover this, we also have a business on nanomedicine for development and cooperation. If you visit our webpage, you can also get the information about the nanoparticle. Regarding our chip, we submitted application for patent, and we also applied for the design patent. If you are interested in this, or if you need more information, please contact us via our webpage, or you can contact us using this contact point. Thank you very much. This is the end of my presentation.
장기 모사 칩의 응용에 대해서 좋은
So
발표해 주신
thank you very much for
CTO
the presentation on the application.
다음 연사분을 소개해드리겠습니다.
And now let me introduce the final presenter and Jin Song-hwan from TNR BioFab. He serves as director. And Song-hwan Jin has a PhD in mechanical engineering from Seoul National University, and he is a professor of mechanical engineering at Tech University of Korea and a director of 3D bioprinting research center in TNR BioFab. And he will talk about 3D printing, and he will share thought-provoking insight with you. So please give him a big round of applause. Good evening. As introduced, I am Jin Song-won from TNR BioFab. So let me brief you on the company profile and our company's technologies will be delivered, and of which I will talk about the preset bioprinting technology, which is very relevant to this session. So TNR BioFab is our company name, and actually we are aiming for tissue engineering and regenerative medicine and biofabrication, and this is a short combination of these three words. And we have been established in 2013, and also we start R&D and commercialize 3D bioprinting platform. And actually we have commercialized 3D bioprinting platform and its applications for biomedical use. And also we are selling this bioreabsorbable scaffold, and also we utilize biomaterials to develop a adhesive patch for immuno-oncology therapy. And in 2018, we were listed on Korean stock market or Kosdaq market. And our core technologies are like this. Actually, when we talk about our technology, three parts consist our technology. This is all about material, and the second is 3D printing system, and third is about process. There are three categories. So when it comes to biomaterial, our company is utilizing tissues, organs of animals, and derivatives of animals. We extrude meaningful substances from this animal, and extracellular material ECM is extracted. So we utilize this ECM from tissue of animal, and we use this to for 3D bioprinting. And this biomaterial, actually I am not including this data, but stem cell technology or cell production technology are combined to produce bio ink, and this bio ink will be used for 3D printing, and the final products or tissues or organs are produced. Sorry for my mistake. And actually this 3D printer we have, before our establishment, the founder of our company was starting their research by creating 3D printer, and all 3D printer are all made on our own. It's a proprietary. Bio ink or biomaterial and 3D printer are well integrated to produce certain tissue or organoid. And with the we also explain this technology as a platform technology. With the usage of platform technology, we have produced 3D printed scaffolds, biosurgical solutions, and 3D engineered tissue equivalents. And also we are doing R&D to utilization in the clinical setting. And we have completed some parts of product line and partial completion of some product line. And also our product production might require more time and effort. So mainly I will talk about the methodology and platform technology that are required for 3D engineered tissue equivalents and therapeutics. So as I mentioned before, 3D engineered tissue equivalents, let me touch upon the concept of 3D engineered tissue equivalents. This is why I prepared for this slide. Before, a few years ago, actually we explained this kind of product as organoid. And this lead to a backlash from the organoid community. And we are sharing the ultimate goal with the organoid community, but actually there is a difference in approaching strategies. So organoid is well elaborated. Actually, it starts from single stem cell, which will be cultured to be proliferated and differentiated to create a final structure. This is organoid structure, but we do not start from one single cell. Actually, we start from multiple cells. So we have to use 10 to 16 power of cells for a small test. So we try to create and control the shape. And with this structure, we culture this and we nurture this tissue, and then will be utilized. So actually, we can be able to control shape. So compared to the normal approaches, in terms of size and shape are easily controllable. And also the short culture time is one of our technology's benefits. So we have 3D tissue. We have produced 3D engineered tissue equivalents by using 3D bioprinting, and there are a garden variety of 3D bioprinting, but we utilizing extrusion bioprinting methodology. And extrusion-based bioprinting, actually we use pneumatic. This is pushing bio ink to the nozzle with the air pressure. And we use pneumatic extrusion bioprinting. This will allow us to use high viscosity bio ink, and for certain structures or high resolution structure or precise structure requiring the small nodule size. This is why the bio ink is ejected the nozzle, and during the process, the low Reynolds number flow is witnessed. Actually, low Reynolds number is, you don't need to know this, but one of the characteristics is the microscopic mixing. There's no microscopic mixing. This is one of the representative characteristics of low Reynolds number flow. And this picture well encapsulates the characteristics of the number flow. And this is not an image, this is a real picture. And there is a picture that encapsulates a small process in a very small environment. And when we let those different colors flow, and from your perspective, colors are mixed, but with low Reynolds number flow, we can identify and distinguish the different colors of fluids. So in your daily life, this can be easily observed by toothpaste. So rather than one color toothpaste, there is a different colors of color liquid in toothpaste tube, and you can see there is a feel in toothpaste with the same structure. So this leads to, actually this can be featured with low Reynolds number flow. So actually there is a noise around here. So this is actually mentioned before, a characteristic of low Reynolds number flow, which will be incorporated into our bioprinting method. This is called 3D printing based on extrusion pneumatic. So we do not start from the small size, we do start from big size. Precursor cartridge is used so that we can fill the bio ink into the desired site, and we let this bio ink into the nozzle. This bio ink is not mixing, so there is no microscopic mixing. This is a basic concept of pneumatic extrusion bioprinting, and this methodology can be realized, and there are 4 different inks used to identify the concept can be really brought into reality. So this is a 9mm precursor cartridge, and at a minimum level of 200 micrometer nodule flow, and there can be a line that is similar to 200 nanomicrometer. So actually we draw 200 micrometer line, but we can also establish a structure. So compared to the normal 3D bioprinting structure, we can create and realize a smaller size of structure. With the technologies that I've mentioned before, we have been committed to attempts to create structures seen in human bodies. So we create cartridge and fill another bio inks, and we also eject this bio ink. And actually of course there's no 100% biomimicking, but we are able to biomimic the structure or organs that we are eager to construct. And also the cell viability can be maintained at a higher level with the utilization of our technology, which is because normally when we do bioprinting, the viscosity high bio ink should be used, and small size nodule should be used. And also there is a shear stress in the wall of nozzle. So there is a nodule wall cell which leads to mutation of cell, and actually this can be stretched and there will be a external force applied. So because of the external force, cell mutates and cell is damaged. So surrounding the nodule wall, the viability of cell is very lower than that, and the cell centering around the center. And the preset bioprinting technology allows us to utilize the bigger nodule to create a smaller structure. So under the assumption of creating same structure compared to the normal bioprinting, we can increase the cell viability. And actually you can see the 4 pictures here. There are 4 divide images, and when you use this bigger nodule, the cell surrounding the nodule wall are dead. So we have to utilize a smaller nodule to increase the cell viability surrounding the nodule walls. So we can prevent or decrease the possibility of cell apoptosis surrounding the nodule wall. So we can increase cell viability with the utilization of our technologies. And also we can create and biomimic human scale liver lobe capillary, as you can see. And we only use endothelial cell only, but we also can find that this can be leading to the perfect construction of desired organs. And we also measure functionality, and without creating structure, we do mixing hepatocyte cells and homogeneity. Compared to this, actually, we can see the increase in functionality when we do structure, when we establish structure. So when we can create bulky structure with our technology. So there are bulky tissues that are very stable in their patterns, and this tissue can be found in liver or neurons and muscles. So we are working hard on developing such tissues. And actually we can draw one line from bioprinting, but we can create spheroid. And with the technology used, we can miniaturize pattern small spheroid, and with this spheroid, and the 2 cell will be mixed with spheroid in a homogeneous manner. And when we compare the 2, in terms of this spheroid, the structure can be maintained a longer period, and also the spheroid form can be well managed and maintained. This is not because of the structure, but actually endothelial cell is encapsulating this structure. Then this leads to a longer maintenance, but without encapsulation, the circle spheroid is not maintained, and this leads to mutation. And when we try to confirm this is implantable, and we do animal study, and there are structured part and non-structured part, and compared to this, we are able to see in vivo implantation of structured one. So 3D bioprinting technology or biomaterial stem cells and operation technologies are all used to provide 3D engineered tissue equivalents, which can be contributing to the drug discovery. We are on the continuous R&D path, and I hope you can see soon. Thank you for your presentation. Thank you. And without further ado, we will move on to the panel discussion. 10 to 20 minutes will be allocated. So in the panel discussion, we are talking about the challenges and limitations of biomimetic technologies, and also ways to weather the difficulties. This will be the topic of panel discussion. And actually, if you really do have a question before you grab your dinner, we will take your questions from the floor.
I hope this panel discussion can be a meaningful time. So please engage in the panel discussion. Then from now on, we will begin the panel discussion. Right next to me, we have Mr. Yu Jong-man from Organoid Science. Using organoid, now in order to be successful in business, what kind of challenges do you have, and what kind of strategy are you implementing? Professor Yu, please go ahead.
And actually, utilizing organoid is on areas of witnessing research activities, but one of the biggest issue is limitation. And when we do produce organoid, there is a variation in that process, and this is a grand issue. And second issue is about vascular issue. So organoid can biomimic the single tissue, but in terms of vessels, or as I mentioned before, in terms of the immune cell, there are a variety of tissues and other components and cells. So organoid should be targeting not only the single tissue, but also the consent of the multiple tissue. And third one is organoid is centered around a single organ. So there are three challenges that we have gone through in terms of commercialization. So first one, in terms of variation, we can overcome with a variety of methodologies. As I mentioned before, we can do IPS and EAS to standardize the core technologies. And second, even though variation is witnessed, there might be a clear cutoff in accordance with different QC indicators. And this is what we have introduced to overcome the challenges. And second, when it comes to the vascular issue, actually organoid can produce vessel, and also it can be attached with organoid tissue, or we can inject vessels into the organoid. So actually angiogenesis is important, but after that we have to biomimic the blood flow through that vessels. But when we do biomimic the vessel, but we are not able to let blood run through those vessels, so this is not technically viable in the near future. So microfluidics can be a solution to mimic vessels. This might be a direction. And third, multi-organ related issues. Actually, organoid is not enough to produce multi-organs. Of course, there might be a flow for creating by creating multiple organoids. However, this is not a silver bullet. So microfluid is, as you mentioned, the microfluid mix and organoid should be incorporated and integrated. This is ultimate direction that we have to pursue. And so perfect combination between the two with advantages will allow us to increase and biomimic the human organoid with a better quality. And this is the ultimate goal. This is the prerequisite for genuine commercialization.
유준만 대표님이 말씀하신 거는 이제
Thank you, Mr. Yu.
organoid의 사업화의 단점을 극복하기 위해서
In order to overcome the challenges in commercialization,
장기모사치과의 융합,
micro 3D organ on a chip should be combined together. So after today, since we are in the same industry, I hope we can contact more frequently so that we can have a new system that can integrate the organ on a chip.
좋은 말씀 감사합니다.
Thank you very much for the good answer.
그다음에는 이제 맵스젠의
And I would like to turn to the CTO of MapsGen, Dr. No.
한계점 어려운 점과
Can you
그걸 극복하기 위한
tell about the challenges
전략에 대해서 말씀
of
주시겠습니다.
the human organ on a chip and the strategy to overcome it? Before organ on a chip, I also studied under the Stephen K. Professor, and he said that he will free from the conventional way. However, microfluidic researchers are now focusing on tubing. So from the end user perspective, how much we can conveniently use this is important because this is a new technology. Even though it is a good thing, even if we want to introduce this for the researchers or for the operators, if they are majored in this, there might be difficulties. So to overcome this problem, we need to simplify this or we need to automate this. So we are also making efforts to automate this. Just like Nespresso, if we inject the cell and push the button, then everything will be done. That will be the ideal scenario. But nowadays, we need to have a separate connection process. So that is the current situation. And in the future, we need to do automation and we can enhance the user friendliness. And another aspect is when it comes to the organ on a chip, how much reputation or approval can be given. Even if we have a good platform, if it is to used in the hospital, we need to get the approval. But this is a new technology, so there is no precedence. So large CROs need to acquire this technology so that we can eventually go to the approval process. Now in Korea, we need to go through the approval, verification, and confirmation. And I believe there is a program and the government is providing the support. So no matter how this is great, this is not a pre-made product. If we are to use this in the medical scene, we need to go through these processes. So Dr. Low mentioned this. In order to be successful in a business, user friendliness is very important. So you pointed out that. And in order to be successful in the business, we need to have standardization and change of regulation. Nowadays, we need to go through the animal test to go to the IND. Legally, we cannot use the organ on a chip to go to the clinical test. However, if many companies have more data showing that it is similar with the animal test, then within several years, I believe we can replace the animal test with organ on a chip, and we can change the guideline. So by 2035, the EPA of the US said that they will not sponsor the animal test, and animal test will be banned. So we need to find a way to go through the clinical study through the organ on a chip.
So let me turn to the Dr. Jin in terms of challenges of 3D bioprinting. So could you please share your take on this? Actually, 3D printing has gone through the biggest challenge, in which Professor CEO Yu said actually angiogenesis is the biggest issue. 3D printing can create a lot of tissues and organs and structures, but we have to enlarge this structure, which requires angiogenesis so that blood flow can be well maintained through these vessels. However, angiogenesis with structures with these vessels properly functioning are not nowhere to be seen via 3D bioprinting. So 1 centimeter by 1 centimeter by 1 centimeter, culturing this is presented as a challenge. So this kind of tissue can be easily produced, but without vessels, it leads to death because of the lack of vessels. It is not able to be maintained longer. So bioprinting technology, when you ask this question to other those in bioprinting industry, everyone says angiogenesis is a problem, and another problem is that a securing cell is very difficult. So a small amount of cell can be achievable, as I mentioned before, 10 to the power of 16 to 17 actually should be acquired for one test. So this large amount of cell should be secured, which leads to 1 to 2 times per week. So if there is a one supplier to produce a cell, we can continuously carry out experiments on a daily basis, but this stands out of our way. And the cells that we are using are not primary, but if we try to use functional cells in the future experiments, the securing cells becomes more difficult. So functional cells should be securely at a large scale, and this is a prerequisite for us to commercialize this. So thank you very much for your answer. And on the floor, from the floor, if you do have a question, so if you do raise a question before leaving this room, please raise your hand before you grab a dinner. Questions can be to anyone. So think your question will be highly appreciated. If you raise your hands, we will deliver you a microphone.
Is there any question? If there is no question, then I believe you want to have dinner. So we would like to conclude the session here. Thank you very much for staying with us until the end of the session, and I wish you have a good return to your home.